Boa noite a todos e a todas. Muito obrigada pela presença de vocês aqui no terceiro dia do nosso segundo Simpósio Pernambucano de elegans. Hoje vamos encerrar o nosso simpósio com um dia incrível, uma noite incrível, com palestrantes incríveis aqui participando do nosso evento. A gente dá as boas-vindas a vocês, agradece mais uma vez por vocês estarem aqui nos acompanhando até hoje. Agradecemos também o nosso patrocinador, a Sanar Saúde, que colaborou conosco com os brindes que a gente vai sortear hoje na palestra, lá no encerramento do nosso evento. E eu gostaria de chamar hoje aqui uma pessoa muito especial, que é o Elias. Olá, Elias, boa noite.
Olá, Priscila, boa noite. Boa noite também aos telespectadores aí do SIMPECE, né, do segundo Simpósio Pernambucano de elegans. Eu, também, que estou muito feliz em estar aqui com vocês, presença de Priscila, que também é uma grande parceira também, já de colaboração, de várias colaborações que nós temos tido. Que tenhamos uma ótima noite, Priscila.
Oi, Elias, eu estou tão feliz que você está aqui com a gente. Elias participou da primeira edição da LAES e hoje ele está na Laica, é acadêmico aqui da Universidade Federal de Pernambuco, do curso de Biomedicina. E eu queria que tu falasse um pouquinho sobre a tua liga acadêmica, Elias. E obrigada à tua liga por ter permitido a tua vinda aqui na nossa LAES.
De fato, Priscila, então, a LAICA, que é a Liga Acadêmica de Imunologia Celular e Aplicada, ela se baseia muito na LAICA, a cadela russa que foi ao espaço, porque ela busca justamente explorar. E explorar o quê? Os campos da imunologia. A gente sabe que no Brasil é também um dos campos que vem crescendo muito, assim como o campo do C. elegans também, dos estudos com C. elegans. E a imunologia ela abrange muitas formas de estudos científicos que nós também pensamos em trazer para a nossa liga. Então, nós estamos também promovendo alguns eventos, alguns tipos de encontros que levam também a fomentar mais esse conhecimento científico, e esse agora no campo da imunologia.
Muito bem. Depois você deixa o Instagram da liga de vocês para o pessoal seguir, acompanhem eles, porque as palestras são excelentes também. Todos os eventos são maravilhosos. Semana que vem vocês também têm um evento, né?
Isso, que é o primeiro simpósio de imunologia celular aplicada. Nós vamos ter palestrantes muito especiais também e contamos com a presença também de vocês.
Muito bem. E Elias também trabalha lá no LICA, no Laboratório de Imunopatologia Keizosami, então a gente sempre se encontra por lá. Muito bem. Elias, vamos então chamar o nosso primeiro convidado e também mediador dessa noite.
Isso.
Boa noite, Hugo. Tudo bem?
Boa noite, Priscila. Tudo ótimo. Obrigado pelo convite. Uma honra estar aqui com vocês.
Seja muito bem-vindo ao nosso simpósio. E Elias vai fazer a tua apresentação para que o pessoal que ainda não te conhece possa te conhecer e saber sobre o trabalho brilhante que você tem realizado aqui no nosso país. Muito obrigada mesmo.
Obrigado.
Isso. Olá, professor Hugo, doutor Hugo, boa noite, é um prazer conhecer o senhor também, de forma virtual, infelizmente não é presencialmente, mas eu acho que é esse um dos prazeres também do virtual, um dos poucos, nesse momento pandêmico, que é justamente essa conexão que nós podemos ter, o senhor está no Rio, eu estou aqui em Recife, e nós estamos aqui no primeiro simpósio Pernambucano de elegans. Então, pessoal, o Hugo, o doutor Hugo Aguilanil, ele é geneticista de formação. E depois de concluir o doutorado na Universidade de Gotemburgo, lá na Suécia, ele fez um pós-doutorado no Salk Institute for Biological Studies, lá nos Estados Unidos da América. As contribuições científicas do Dr. Hugo se deu principalmente no estudo das bases genéticas do processo de envelhecimento. Ele estuda a longevidade, envelhecimento, nutrição, reprodução, e basicamente esses estudos são feitos no nosso modelo querido, que é o Caenorhabditis elegans. O Dr. Hugo também faz parte do Conseil National de la Recherche Scientifique da França, onde é também diretor de pesquisa. Lá, ele liderou uma equipe de pesquisa na École Normale Supérieure de Lyon, e a pesquisa dele foi premiada, tanto pelo Conselho Europeu de Pesquisa, como também o francês, Dr. Hugo. Em 2017, o Dr. Hugo ele se tornou também diretor-presidente do Instituto Serrapilheira, que é uma instituição sem fins lucrativos que fomenta a ciência brasileira, de forma a estimular o pensamento crítico, científico e também criativo. Ele também é membro do Conselho Superior da FAPESG, e este e muito mais é Dr. Hugo Aguilanil.
Obrigado, Elias. Que apresentação? Tá lindo, mas é isso mesmo. Muito obrigado, o prazer estar aqui. Eu estou duplo prazer como diretor do Serrapilheira e como pesquisador de elegans, né? Eu acho o que vocês estão fazendo aqui uma coisa fantástica, na verdade. E faltava um meetings de elegans no Brasil. Pernambuco que está saindo na frente fazendo isso, eu acho fantástico. Eu acho que isso é uma cultura de elegans, né? Cultura de se encontrar, cultura de comunidade. Tem um grande meetings de LA, né? Que esse ano infelizmente foi virtual, mas eu acho fantástico. Isso é uma coisa que dá para fazer, dá muito prazer nesse momento muito crítico do Brasil, né? De ver iniciativas assim, cheio de gás, de energia, convidando pessoas do mundo todo para falar sobre esse elegance. Eu acho fantástico. Vocês estão de parabéns.
Muito obrigado, doutor Hugo. Aproveitar os momentos também, né, que tem acontecido, né, para a gente poder formar aí as conexões, né? Eu acho que é importante esse tempo complicado que o Brasil tem vivido, é formar conexão, é formar campos de ajuda, né? Eu acho que é o momento que a gente vem para fazer isso. E para começar falando então sobre isso, né, eu queria trazer, rebuscar do seu uma memória. Eu estava assistindo ontem ao seu TEDx, na Conferência de São Paulo, lá na USP, e aí tem algo que você falou muito importante, que eu acho que é sobre intuição no campo científico. O senhor fala, o senhor equipara, na verdade, nós cientistas, né, ou iniciantes científicos, que é o meu caso, a artistas no campo da formulação da hipótese, né? No caso, a hipótese teria uma ajuda da intuição. E aí eu queria perguntar ao senhor, doutor Hugo, qual foi ou como se deu essa intuição do senhor que levou o senhor também a usar o modelo que a gente gosta, que é o C-elegans, né? Como é que o senhor viu nas suas intuições, nas suas hipóteses de formulação, como é que você viu que o C-elegans ele se encaixava como modelo experimental para os trabalhos do senhor?
Vamos dizer assim, eu não usei tanto a minha intuição para escolher o modelo, porque já me interessava. Na verdade, eu comecei a estudar envelhecimento usando levadura. Então, minha tese na Suécia não usava C. elegans, usava levadura. E aí, óbvio que isso, uma célula é um modelo muito impressionante também, mas é unicelular, então tem limitações, né? Se você quer entender como um ser complexo, um pouco mais complexa, pode reagir a um meio ambiente, por exemplo, o C. elegans é, obviamente, um modelo muito atraente, né? E também eu sou geneticista, então eu gosto de animais que a gente conhece todos os genes, que a gente pode fazer manipulações com facilidade. O fato de poder usar, quando eu comecei meu postdoc, foi 1 ou 2 anos depois do famoso paper do RNA, né, do RNA com interferência do Mello e do Andrew Fire. Isso, a possibilidade de você poder abaixar a expressão de um gene com RNA de interferência era uma coisa assim, absolutamente incrível, né? Quer dizer, isso abria todas as portas para fazer estudos muito interessantes. Então, quando eu fui para os Estados Unidos, realmente o C. elegans foi o modelo meio que óbvio, né, para mim, porque, bom, estudando um ser que tem uma expectativa de vida curta e que é geneticamente maleável, para mim era o paraíso. Agora, a intuição, ela é importante no desenho experimental, eu acho, né, porque muitas vezes, quando você tem que pensar sobre o próximo experimento, que não é uma coisa simples, aí eu acho que a intuição entra no jogo. Porque isso é uma coisa que as pessoas, às vezes, que dá para ver que as pessoas não percebem muito isso, né? Porque o cientista ele tem essa imagem de ser um ser muito racional, muito... Na verdade, a intuição ela faz parte do processo científico bem no início, né? E não só na biologia. Aliás, os livros mais incríveis escritos sobre a intuição são de matemáticos, né? Os matemáticos, antes de começar uma demonstração, muitas vezes eles têm um pensamento muito intuitivo. Então, por exemplo, eu sempre trabalhei na interação entre a reprodução e o envelhecimento. Isso vem de uma observação, na verdade. Eu percebia que depois do período reprodutivo, aí era o momento que realmente os animais ficavam mais fracos e começavam a envelhecer com uma velocidade muito mais importante. Aí a pergunta será que o processo da reprodução tem uma ligação com a velocidade com a qual o animal envelhece? E se eu finge que o animal não pode se reproduzir, o que acontece? Ou se eu mando o sinal que o animal pode ainda se reproduzir, mas não tem como fazer isso agora. É aí que acontece para o envelhecimento. E essas são coisas assim, totalmente intuitivas. Não tinha dados que me indicava que era isso que precisava pesquisar. Mas é isso que é gostoso, né? Porque você tem como testar suas intuições. Exato. Aí vem o processo científico, que obviamente vai descartar a grande maioria das suas intuições, mas algumas podem dar certo. Então é isso. Exato. Eu acho que é lindo, esse processo é lindo, e as pessoas simplesmente não conhecem suficientemente o nosso trabalho como cientista de explicar como que isso acontece, né? Porque você sonha sobre o teu experimento, você tipo, vai para a cama de noite, começa a pensar. Isso é totalmente intuitivo. Você não precisa ter uma coisa racional. Aí, essa intuição, ela vai te dar ideias e você vai testar o dia seguinte. A grande força, né, da ciência é que você tem como testar suas intuições. Isso é uma coisa incrível.
Exato, e é algo que a gente é parte do simples, né, doutor Hugo? É um pensamento, na verdade, que traz, e que vocês também lá no Serrapilheira buscam isso para colocar nos editais. Vocês buscam que, primeiramente, a gente discorra sobre o simples, né? Como é que você se explicaria a sua proposta, né, de pesquisa de forma simples, de forma esmiuçada, para que a gente possa também elaborar, né, e discutir, né, que eu vou trazer agora uma nova pauta, sobre a divulgação científica, né? O senhor trata aí várias vezes, né, sobre essa importância da gente também desmistificar o cientista, né? Aqui no Brasil, né, pelo menos de vovó fala muito em gente sisuda. Sisuda é uma pessoa que vive, sabe, com o olhar fechado, meio sério. Isso é a imagem que a gente às vezes tem do cientista. Mas aí o Serrapilheira ele traz diversas formas de fazer com que isso não seja bem verdade, né? E aí divulgar a ciência. E aí, E aí, pensando um pouco sobre isso, eu queria perguntar ao senhor sobre o seguinte: como é que o senhor visualiza também esse momento, né, que a gente vive aqui no Brasil em relação à pesquisa científica e à ciência, porque o senhor se insere no contexto brasileiro, digamos assim, em 2017, né, no contexto de ciência brasileira em 2017, quando o senhor assume a presidência do Serrapilheira. E aí, desde essa época, a gente já via que a ciência brasileira começava a estar em trancos e barrancos, como a gente fala por aqui. E aí, agora, você se encontra no olho do furacão, praticamente, né, da ciência brasileira. Então, como é que o senhor visualiza, né, essa importância de se investir na ciência brasileira? Para o senhor, ainda vale a pena?
Claro. Senão não estaria aqui, né? Claro que vale a pena, mas isso dito, eu acho que a situação é grave. Eu acho que a gente está passando por uma situação muito grave. O Brasil é um dos poucos países que, normalmente, quando os cientistas sempre reclamam para ter mais financiamento. Normalmente, o que a gente reclama é que a velocidade de crescimento do investimento na ciência pode ser acelerado ou reduzido, mas sempre há um crescimento, né? O Brasil faz parte dos pouquíssimos países onde tem uma queda, né, em comparação ao PIB. O tanto de dinheiro que foi investido ano passado ou 2 anos atrás é mais que esse ano. Isso é gravíssimo, né, porque isso interrompe, claro, o processo, né, muita gente fala, não dá para você desligar e ligar novamente, não é assim, o processo científico é uma coisa que você tem que ter continuidade. Então, mais que isso, acho que ideologicamente nós estamos numa situação muito, muito diferente, muito peculiar, com um governo que nitidamente tem um problema, vamos dizer assim, para ficar suave, com todas as atividades que geram conhecimento. Então, ciência faz parte dessas atividades, né? São as atividades que podem levar o país a crescer. Cultura, arte, ciência. E nitidamente nós estamos numa situação que há uma descredibilização, né, das instituições. As universidades federais estão numa situação muito ruim, o CNPq também está numa situação ruim, e obviamente os pesquisadores são pessoas inteligentes, então, eles entendem que o ecossistema federal, vamos dizer assim, não é favorável, né? Agora, isso não vai ser para sempre. Então, eu sei que é difícil dizer isso. O Serrapilheira não tem o tamanho suficiente para se substituir. Muita gente fala para mim, mas vocês podem entrar, agora que o governo não está mais. Mas, assim, as pessoas não têm a noção do tamanho do investimento que o governo representa. Ele não se compara com o Serrapilheira. O Serrapilheira tem um orçamento anual que é muito pequeno em comparação com o CNPq, a CAPES, e o mesmo a FAPESP, a FAPER, não é o objetivo do Serrapilheira. O Serrapilheira, na verdade, ele tem que funcionar acima de um sistema público robusto, né? Então, se não tiver um problema, uma política científica robusta pública, e ela tem que ser pública, não tem como ser diferente, o principal do investimento tem que ser público, e sempre é assim, qualquer país digno, é assim que se faz, né? Aí você pode ter financiamentos anexos, como o do Serrapilheira, que abrem uma outra possibilidade. Há uma possibilidade de fazer projetos um pouco mais ousados, de você articular, financiar divulgação científica, mas se não tiver um ecossistema robusto atrás, isso não serve para nada, né? Então, obviamente, o Serrapilheira se preocupa muito com a situação atual, mas eu também acredito que essas situações muito adversas, elas são propícias à construção de algum tipo de resistência, né? E, quando a situação é muito ruim, sempre cresce uma florzinha ali e lá, e isso vai dar a base para o próximo passo, né? E o que vocês estão fazendo de uma forma é isso, né? Quer dizer, eu acho lindo que num momento tão crítico, há esse simpósio do Pernambuco sobre esse elegans, com pessoas assim, sorridentes, cheios de gás, interessados, querendo fazer ciência, organizando coisas sabe? Isso eu acho que é a base do amanhã. E, obviamente, a gente tem que trabalhar para que os cientistas sejam mais visíveis, a ciência seja mais visível, porque ela está, por causa da pandemia, no meio do debate público nesse momento. E a gente tem que trabalhar para que ela fique um tema central, né, de políticas públicas. Porque o próximo governo vai ter que se engajar a apoiar a ciência. A ciência mostrou uma eficácia incrível durante a pandemia, né? Então, a gente vai ver com o tempo, obviamente, as pessoas se concentram muito sobre os pequenos debates que houve, que, na verdade, o próprio processo científico, ele é cheio de debate, isso que faz a ciência, mas o grande da produção científica é que há vacinas, né? Simples assim. Quer dizer, essas vacinas, elas são o resultado de um investimento de vários anos, de vários países, e é incrível que, com tanto pouco tempo depois do início da pandemia, a gente já está com vacinas que funcionam, e não só uma. Então, isso é simplesmente a comprovação mais incrível da necessidade, da utilidade do lado estratégico da ciência. No Brasil, além disso, tem a questão ambiental, né, que não vai se resolver sem uma ciência forte no campo da ecologia, em particular. Mas, enfim. Então, eu estou preocupado, mas eu estou cheio de esperança, e o que me dá mais esperança são exatamente iniciativas como essa. Pessoas. Eu vejo que tem muita gente no Brasil que são absolutamente incríveis. Então, eu acredito muito nisso. Você falou uma coisa interessante, Eli, é que esse período realmente ele permitiu, de uma certa forma, uma conexão do Brasil com o resto do mundo, né, com essa coisa virtual, que eu acho que é uma coisa que o Brasil tem que fazer cada vez mais, né? O Brasil, às vezes, se sente um pouco longe, um pouco difícil de colaborar, e não é. Você vê, hoje nós temos speakers que vêm da Alemanha, dos Estados Unidos, e as pessoas adoram participar. Eles, sabe, não é que o Brasil é isolado. Então, isso talvez seja uma coisa que a gente possa ampliar, né, no futuro. Eu acho que o Brasil precisa colaborar mais, precisa trazer pessoas de fora. Tem muita gente que quer vir para o Brasil, que quer colaborar com o Brasil, porque o Brasil é um lugar muito incrível, né? Então, eu acho que não é uma coisa absurda de se pensar.
E aí, eu concordo absolutamente com tudo que você falou, e aí eu trago também, né, já que você falou também sobre o tema que eu já ia comentar, que era sobre essa questão das conexões, eu queria entender do senhor também sobre, agora a gente vai olhar para um outro Hugo, que é o Hugo que revisa periódicos. Então, o doutor Hugo que revisa periódicos, né, revisor de periódicos de grande fator de impacto, né, de impacto científico. E aí eu queria linkar duas questões que no Serrapilheira vocês abordam o impacto científico de forma diferente do que só números, não é verdade? Você quer falar um pouco sobre isso?
Exatamente. Não, totalmente. Não, eu acho que, bom, a gente está no momento de transição, né? A ciência, o sistema científico em geral. Então, a gente quer muito pensar cada vez mais sobre ciência aberta, né? E ciência aberta, isso é uma palavra simples, rápida, mas o que está atrás disso é muito mais profundo, né? Quando você diz que você quer que os dados sejam abertos, isso, como que eles vão ser revisados? E como você é um jovem cientista, obviamente, você vai querer publicar na Nature, na Science. Esses jornais não são abertos. Então,
Exato.
como fazer? Agora tem planos, né? Por exemplo, tem um plano S, que as instituições de financiamento dizem: a gente vai pagar um preço a mais para que cada artigo na Science ou na Nature ou qualquer jornal desse tamanho seja aberto. Será que isso é a solução? Pagar mais os jornais, que já recebem muito de instituições públicas até? Certamente não. Então, como a gente tem que repensar o sistema científico inteiramente, né? E o que conta no final não é o fator de impacto, não é o jornal, não é nada disso. É a descoberta. O que está no paper, né? Concretamente. Isso não é uma coisa nova, mas o sistema científico, infelizmente, com o sistema editorial, e não é a culpa dos editores só, mas certamente isso contribui, foi um sistema meio que de oferta e demanda, né? Então, tem jornais que ficaram muito grandes, e esses jornais agora têm um poder que é demais, que é grande demais. Eles são caros. E, obviamente, quando você trabalha na Harvard, o preço de um artigo não é um problema. Agora, é um problema para outros pesquisadores, e a gente sabe que talentos não têm geografia, então você pode ter pesquisadores incríveis na África, em países muito, muito desfavorizados, e para eles, pagar $5.000 para publicar um artigo é simplesmente impossível. Então, você aceitar que vai ter uma segregação violenta no mundo do conhecimento, que deveria ser um mundo mais aberto possível. Então, essa reflexão, o Serrapilheira realmente quer participar dessa reflexão no Brasil. O Brasil é um país que já é avançado na questão da ciência aberta, né? O sistema Cielo foi um dos primeiros sistemas abertos. Então, isso é uma boa. Mas não é só colocar uma plataforma, é muito mais que isso, é repensar. O Brasil tem que repensar conjuntamente com os outros países o sistema científico. Então, a gente realmente, isso é uma questão que é importante. Na nossa seleção, nós fazemos questão de não tomar em consideração o fator de impacto.
Uhum.
E aconteceu muitas vezes, eu posso te dizer isso, na nossa avaliação científica, que um cientista com papers incríveis, com um fator de impacto muito grande, não passou da seleção simplesmente porque não estava claro a contribuição dele, a contribuição intelectual, a pergunta que ele estava fazendo. E assim, não é obviamente, quem consegue publicar nessas revistas muitas vezes são pesquisadores muito bons, mas não é sistemático. E aconteceu várias vezes que um pesquisador que tinha uma pergunta fabulosa, mas que não tinha os papers muito incríveis, passou na frente de um outro pesquisador que publicou muito bem, mas que não tinha a capacidade ou não tinha explicado suficientemente bem a pergunta científica que ele estava querendo fazer. Então, isso quem conta, é a pergunta, é o problema, e como você vai resolver. Isso que isso é no mundo dos elegans, o Sidney Brenner, que é um dos grandes, o pai, né, da verdade, do campo dos elegans, falava, né, que eu lembro ele estava no Salk Institute, né, e eu lembro que ele quando jovens cientistas chegavam para ele, a introdução sempre é a mesma. Ele publicou na Science, ele publicou 3 artigos na Science, 5 artigos na Nature. Eu não quero saber aonde ele publicou, eu quero saber sobre o que ele publicou. Ele pode publicar em qualquer jornal, isso não pouco importa. Aliás, o Sidney Brenner ganhou um prêmio Nobel, nem com o Science, nem com o Nature, ele publicou muitas vezes lá, mas o paper que deu para ele o prêmio Nobel é um paper na Genetics, que é um ótimo jornal, mas que não é também esses jornais muito grandes, onde simplesmente ele caracterizou o C. elegans, deu todas as condições para trabalhar, e o campo inteiro foi feito em cima desse paper. O cara é um genial. E ele não prestava atenção no fator de impacto. Então, é um bom sinal.
Exato. E é sobre isso que a gente tem tentado também abordar aqui na Liga, né? Falar um pouco sobre ciência, não, como posso falar, não vermificada, mas humanizada. E a gente tenta também, então, trazer isso agora com a doutora Júlia Bornhorst, que vai trazer um pouco, então, sobre as perspectivas, né, científicas que ela tem trazido lá na Alemanha. Doutor Hugo, eu passaria com o senhor muito tempo conversando facilmente, porque eu sei que o senhor é bom de falar, eu também sou bom de falar, mas infelizmente nosso tempo encerrou, e aí eu vou passar agora a palavra e apresentar a doutora Júlia. Muito obrigado e ótima noite para o senhor.
Prazer, um grande prazer estar aqui com vocês, uma honra, aliás.
Uma honra nossa. Um abraço. Hello, Dr. Julia, are you here?
Hi, Elias.
It's a pleasure for us to have you here with us, Dr. Julia. I know that you are a friend of Priscilla since a long time. And so as we are in friends stage, I'm going to present you to our attendees, okay?
Thanks.
So good evening, everyone that is watching us. It's a great pleasure for me to introduce our next speaker today. That is Dr. Julia Bornhorst, and she's going to talk about the zinc and manganese interaction in nematode C. elegans. Our speaker studied food chemistry at the University of Münster in Germany, where she also did her PhD working on manganese-induced neurotoxicity. After, she worked as an independent postdoctoral researcher in Vanderbilt University Medical Center in Nashville in the US. She worked in the field of alternative animal toxicity, testing by establishing new testing strategies for C. elegans. In 2019, she received the Early Career Award in 2019 for her contributions in the field of environmental mutagenesis, and currently she is professor in food chemistry at the University of Wuppertal in Germany. Welcome, Julia Bornhorst, Dr. Julia, and have an excellent presentation.
Thank you so much, Elias, for the very kind introduction. I'm getting a bit wrapped, as you already see. Thank you so much. So I think you already see my slides. I hope so, and you can hear me well. Otherwise, please complain.
We are
I hope
hearing and watching you.
I hope that everyone can is in a good mood. So good evening, everyone. And I just say, in case I'm like too fast or you have any things which you are concerned about, please ask later any question you have. Stupid questions are not existing, and it's a pleasure for me to answer all your questions. And in case your questions are in Portuguese, I think Elias will be in a good mood to be a dolmetscher to translate. So thanks, first of all, to Priscilla, Diana, and all of us who invited me and to gave me here the opportunity to present some of our recent findings. So today we want to focus on trace elements. So I want to give you some insights in what we found in case trace elements are oversupplied. So oversupply, like the organism is taking up too much of the trace elements and we have adverse effects. And also I want to give you some insights into interactions of trace elements. So, but I will come to that later. So let's start first of all with why are trace elements important for us and where do they occur? So the big four, all of you are familiar with them. The big four elements are the main elements of the body. And also most of you are familiar with the major elements, as for example, calcium or sulfur or sodium. Most of you already know them. And then we have the group of the trace elements. It's a group of 13 elements, and in total in the body, they have an amount of 4.5 gram. And from these 4.5 gram, it's very interesting, 3 gram is covering only by iron. So you see that it's only a small amount in the body we have, but they are very essential for life. So it's very important to take up enough from those elements. And we know it's very important not to take up too much of these elements, because otherwise we also have an adverse effect for health. Normally, we cover all the requirements of the trace elements by drinking water and eating food. So most of you cover all the elements. So as I already introduced, it's very important that you maintain your trace element homeostasis in the body, because otherwise it may be a risk factor for a lot of different diseases. So a deficiency is a problem, but also an oversupply is a problem. So for most of you know iron. In iron deficiency, people feel tired or have other health issues. In case of an overload with iron, people have a risk for neurodegenerative diseases, as for example, Parkinson's disease. So you see how important this is to be in the homeostasis. And even if we know it's very important to maintain the homeostasis, and we know deficiency and overload is a problem, we still are struggling with really understanding the underlying mechanisms, why trace elements are causing adverse effect in case of oversupplying them. And further on, what a lot of people don't know, we also have not enough suitable markers to access the trace element status in the body. So it's not 100% clear if the recommendations are valid. And also, we have no recommendation, for example, for an age-specific intake or maybe a disease-specific intake. So all of this is pretty unknown. And you see, we have a lot of gaps in the trace elements. And we have another further gap. So we have a gap regarding the genetics. So what do I mean by genetics? You know that Parkinson's disease, for example, we have 11 genes identified, and we know that in case we have a mutation in one of those genes, we have a higher risk for Parkinson's disease. But what we don't know yet is if a genetic predisposition is maybe affecting the trace element status, or if those patients or models are hyper or hyposensitive towards trace element exposure. So you see, we are really at the tip of the iceberg, and we have to identify a lot of different roles, especially also in the genetics. And to answer a lot of these gaps, we are using C. elegans as a model system. I think in this community, there's no need to introduce the advantages of the model system. I only want to highlight we have a fully sequenced genome here. So we do have a lot of different disease models we can work with. And the good thing is also the trace element homeostasis are fully conserved in this organism. So we can take the organism in order to study trace element homeostasis, also consequences of deficiency and overload. But we will focus on the overload today. Before I want to start and to talk with you about interactions, I want to present you some data we got in the last years regarding adverse effects of single elements in case we are oversupplying them. So we want to focus today on the trace element manganese and the trace element zinc. For manganese, we have now worked for more than 10 years. So manganese, all of you know, is an essential trace element. It is essential for normal brain and neuronal function. And due to its ubiquitous presence in the environment, the daily uptake is higher as compared to the estimated daily requirement. In consequence, a manganese deficiency is very rare in humans, but an overload is a concern. An overload of manganese can cause permanent neurodegenerative damage, and the phenotype which can be observed here is called manganism. It is highly resembling symptoms observed in Parkinson's disease. But still, we don't know the underlying mechanisms of manganese-induced toxicity. And we work now, as I told you already, for more than 10 years in this field. And we used to answer some of those questions in vitro, so cell culture systems, but also the worm. From cell cultures, we could learn that manganese overload is causing neurodegenerative damage. And we could identify by using cell culture models that the blood liquor barrier might be the major route for manganese into the brain after oral intake. But I don't want to go into detail on the cell culture studies because I want to show you some data today regarding the worms. And the worms gave us important hints about potential underlying mechanisms of manganese-induced toxicity. And here we could identify manganese-induced reactive oxygen and nitrogen species, short RONS. We observed an effect on the genomic integrity, and also we observed that in case we do have a Parkinson's disease model, we do have a different manganese homeostasis. To study the in vivo mechanisms, we focused first on manganese-induced oxidative stress. First of all, what is oxidative stress? Oxidative stress is a dysregulation or a dyshomeostasis between the oxidants and the antioxidants. So an excessive amount of free radicals occur, and those excessive free radicals may damage macromolecules and other structures in the cell. An excessive amount of radicals is associated to a lot of human diseases, and this is all covered by the term oxidative stress. So It's not easy, as most of you know, to measure oxidative stress since we have highly reactive species, and we have to take into account that maybe during sample preparation we are also causing oxidative stress or oxidative damage. So we developed a lot of different methods in order to detect in vivo oxidative stress in the worms. We had developed some dye-based assays, but also some chromatography-based assays in order to have here a high sensitivity and a really sensitive method to identify endpoints of oxidative stress. You see here in the graph that we could, by dyes, identify reactive oxygen and nitrogen species in alive worms, and at the lower bottom of the slide you see that we also could identify mitochondrial-derived RONs. What we also could observe is a depletion of the ATP level in case worms are oversupplied with manganese. We could identify a shift towards the oxidized form of the glutathione, and we could identify damaged lipids in the form of the formation of isoprostanes. So I think that all of you are familiar with or are thinking the same. Manganese is causing oxidative stress. Taking now oxidative stress into account, we wanted to know what is now the consequence of such an amount of free radicals in an organism. Because free radicals may affect and damage macromolecules as, for example, the DNA. The DNA, maybe DNA bases get oxidized, or the radicals may induce single-strand breaks. So these were all questions we had. And at the time we started with that, there were only few studies existing looking for the integrity of the DNA in the worms, and only few studies analyzing DNA damages in the worms. So we did some previous studies, and in the beginning we found out, okay, you know, in case you have a DNA damage, it may be repaired by DNA repair. In our previous studies, we found that the L1 stage worms, there the DNA repair is not fully developed. So we decided to work in L4 stage worms in order that the DNA repair is fully developed. And then we had to establish a lot of new methods in order to detect DNA damages in the worm. And we were very successful here, so we could establish a method in which we could quantify the oxidative form of the DNA-based guanine. It's a so-called 8-oxo-DG. 8-oxo-guanine is one of the DNA bases which is very easily oxidized, and it's taken in human samples very often as a marker for DNA damage. So we were able to identify 8-oxo-DG in the nematodes by an ELISA, but we could verify this by mass spectrometry. And what we did further is we developed a method in which we could measure the amount of double-stranded DNA in the worms. Because if you have a reduction in the double-stranded DNA, you have an indicator for DNA strand breaks, and also, consequently, you have an indicator for a decreased genomic stability. And also here, we could find that manganese is decreasing the genomic stability of the DNA in the worms. We were then wondering about, okay, we have identified excessive amount of radicals up in manganese exposure, and we have identified DNA damages which are caused by manganese oversupply. We wanted now further to know, is also the DNA repair affected or activated by an oversupply of manganese to the worms? And for this, we used gene expression studies and looked at all the genes which are involved in the DNA repair. In the DNA repair, we differentiate between different pathways. And the base excision repair is the pathway which is dealing with small base lesions as single-strand break or 8-oxo-DG. So you see here the genes which are involved in the base excision repair, and up in manganese exposure, we saw that especially the genes which are recognizing a damage are upregulated. What you can see in here. So we got a hint for an activation of the DNA of the base excision repair. So I could show you that manganese is causing oxidative stress. We do have an activation on the DNA repair. We have to look if this is a dysregulation or a repair of the damages which are caused by manganese. And we were then further interested in if we know that manganese is activating the gene or increasing the gene expression of the recognition step of the DNA repair pathways. How does a worm behave having a loss in specific of those genes? So we took mutants of the worms lacking these glyoxylases, which are recognizing a damage, and we found a worm which was very hypersensitive towards manganese exposure. So these NTH1 worms died very early. So this is an NTH1 deletion mutant, and they died very early up in manganese exposure. NTH1 is a glyoxylase which is removing, which is recognizing and removing oxidized purines and pyrimidines. Right now, we are still looking for the mechanisms why this worm is hypersensitive. In case you have any idea, I'm happy to discuss this later. In a different study, we were interested, as I told you, genetics might also have an impact, and we were very interested in how do worms behave having a genetic predispositions for Parkinson's disease. So we took worms having a mutation in the Parkinson's disease associated genes, for example, Parkin or DJ1. And what we found was very surprising. So in case we have a loss of the Parkinson's disease associated proteins, this resulted in a higher uptake of manganese and consequently in a higher amount of oxidative stress. So we were wondering, why is only the genetics causing a manganese accumulation in the worm? And here we could, together with a lot of different collaboration partners, identify a novel role of one of the Parkinson's disease genes, the Parkin gene. This is regulating the export of manganese, and due to the loss of function here, the export is not functioning anymore, and this is why you have an accumulation which you can observe then in the worm's body. So you see what impact genetics might have. And in further studies, we went one step further. So alpha-synuclein, it's another hallmark for Parkinson's disease. And in those mutant worms, we integrated the alpha-synuclein with collaboration partners. And by integrating in the Parkin mutants, the alpha-synuclein resulted in a reduction of the manganese level, so it was then comparable to wild type, and also oxidative stress. So you see the potential we do have in genetics maybe are also as a therapeutic strategy. We don't know yet. So with manganese, I could show you a lot of different things regarding what's happening up in overdosing and what maybe underlying mechanisms are. We also focused in the last years on zinc. Zinc, you all know as a trace element, which has a lot of different catalytic functions for enzyme and proteins. And you also know that free zinc ions function as a second messenger. A zinc deficiency is associated with growth disorders or reduced wound healing, or also with a reduced immune function. If you have an oversupply with zinc, it is associated with neurodegenerative disorders. So we were interested in what adverse effects is impacting the worm when zinc is overdosed. And for zinc, you remember that I told you in the beginning that for some trace elements, we do not have a very good marker for the status, and zinc is one of those trace elements. So what is a good marker? The total zinc status in the body, or in the blood, or in the serum, plasma, or is it maybe just the active form of the zinc which is important? So we were looking at this in the worms. So we developed a method with a dye, and this dye is having a fluorescent signal in case free or label zinc is around. And we compared those data with the label zinc and compared this to the total zinc amount in the worms. And we found that these are two different markers. And this was very interesting. And we also found that that we have on both markers an age-specific alteration. And also we used here Parkinson's disease models, and we also could identify here that especially the label or free zinc homeostasis is different as compared to the wild-type worms. So we are right now trying to identify underlying mechanisms and consequences for the worm. As I already told you, we can look at single elements, but this is not the real-life situation. The real-life situation is you take up food. You not take up only manganese or only zinc, or normally you are exposed to both of them. So we were wondering, okay, what is happening in case you are exposed to both elements? And this is part of a research group which is dealing with interaction of trace element in healthy and diseased elderly. So it's a coordinated project where we analyze in aging patients the trace element profiles in order to find out, do we have an age-specific trace element profile, or do we identify a disease-specific trace elements profile? And within this coordinated project, the worms are a very good model in order to study what are basic mechanisms of interactions of trace elements. In order to find out how a worm behaves during aging, we started in the first approach with the question, how are trace elements changing in the worms while aging? And we first aged the worms, and that wasn't that easy as we were thinking, because all of you working with worms know they need every day new food, and they got a lot of larvae. So we found in the beginning a way in order to separate the worms from their progeny, in order really to let them age, and that they have enough food. And then we analyzed at young worms, so day 2 or day 1 old worms. We looked at day 5 old worms and day 12 old worms. And here you can see that we already identified that we do have a change in the trace elements while the worm is aging. Iron is accumulated, but also zinc and manganese is accumulating. So we were asking the question, in case a worm is accumulating with age, manganese and zinc, how would the worm behave in case you are overdosing zinc and manganese the whole life? Does a worm, for example, age more quickly or has a shorter lifespan? These were questions we addressed. So first of all, why manganese and zinc? I already told you they're increasing with age, and both of them have, for both of them, a balanced homeostasis is very important. Both of them are sharing transporters, so they interact in their homeostasis. And this is why we addressed this. And we addressed also the question, if we know overdosing both of the elements, what are consequences in case we have now a Parkinson's disease model? And this I will show you. First of all, I just give you an idea about the study design. So we started to expose the worms with manganese and zinc at L4 larvae. And from L4 on, they got manganese and zinc for the rest of their lives. We took samples at day 2, because these are very young worms. We took samples at day 5, because they are kind of mid-aged. And we took samples at day 12. Day 12 is already the day where 50% of all the worm died due to aging. So we have here a late life worm. So how did we expose the worms? First of all, we determined and quantified how much manganese and zinc is in the E. coli we are feeding and in the agar plate. And then we enriched the E. coli by the amount of manganese and zinc we wanted to feed to the worms. So we fed 5 millimolar of manganese and 2 different zinc concentrations. Very important here, we have done all the experiments in inactivated E. coli. Otherwise, we were not sure if maybe the species are differing or if the E. coli is somehow like interacting with the trace elements. And to exclude that, we inactivated the E. coli. So worms were fed with manganese and zinc in parallel over their lives. What did we analyze? We looked for what consequences do we observe for lifespan, what consequences do we see any changes in the manganese and zinc uptake, and we looked in a gene expression screen for a lot of different pathways which have been identified to play a role in case you expose to one of the elements. Because at the time we started, there was not a lot knowledge about interaction. And I told you that both of the metals, in case they are oversupplied, can cause Parkinson's disease or neurodegenerative diseases. That's why we also took a look at the new generation in the worms. Only that you get an idea. So you have here the normal, you see on the slide, normal dopaminergic neurons, which are GFP marked. You see also splitted somar. This is a very severe damage, so we defined this as a severe damage. You will see later on the slide. We also found some blabbing or some discontinuous signal. These are more mild damages we saw, and you will see this later on the slide. First of all, we checked lifespan. This data are pretty boring because manganese and zinc in parallel and in the single dose does not change lifespan. So we did not have any effect on the lifespan. So next, we analyzed the manganese and zinc uptake in the worms. And you see that in the purple bar, you see that in case manganese is fat, we find more manganese in the worm. In case zinc is applied, in the blue bottom on the right, you see that you have more zinc present in the worms. Very good. Then you see the mixed exposure. So adding manganese and zinc in parallel. And you see that the worms take up more manganese in comparison to just feeding the manganese alone. This was very surprising for us because we did expect a lot, but not that the worms are taking up more in case we are exposing both elements. It was the same for zinc. So very interestingly, and we really wanted to know why is the worm, in case you have both elements present, taking up more of the elements. So we did a lot of gene expression studies for the importers which were associated with manganese and zinc, but the importers were not really differing between the single application of manganese and the co-exposure with zinc. But we found a very interesting effect. So what was really increased in case we are feeding the worm manganese and zinc was the gene expression of the metallothionein. The metallothionein is a protein which is normally more known in the zinc homeostasis. It's a zinc storing protein. So it was very interesting for us that in case you feed the worms manganese and zinc, you have up to 60-fold increase in the metallothionein expression. This special role of the metallothionein we could identify by using worms having a loss in the metallothionein 1. So very impressively what we found here. We then looked further on, do we have any more or severe new generation by feeding manganese and zinc? And we can say here no. So we saw definitely an age-specific or an age-specific phenotype. So with age, the DAT1 is decreasing and the new generation is increasing, but it was not hastened by manganese, zinc, or exposing both. I told you that we also wanted to know how is these effects regarding manganese and zinc together are loaded up more as compared to the single elements. Is this also taking place in case we do have a worm having genetic predispositions for Parkinson's disease? And here we found that in case of a Parkinson's disease mutant, you have an altered homeostasis of manganese and zinc. So the single elements do have an altered homeostasis, but also in case of both elements are present. And the uptake you see here in the green bars where the wild type was, and you see that the mutant worms having Parkinson's disease take up less of the elements. We did a lot of expression studies. Also, the metallothionein has an important role here, but to be honest, the full mechanism we could not identify yet. Very interestingly, in the Parkinson's disease mutants, we found that aging is causing more new generation, but also we can hasten this effect by adding manganese and zinc. So here we have a more severe dopaminergic neurodegeneration upon overexposure. So to summarize, what could I show you? I could show you that zinc and manganese do have an interaction. I could show you that in case they are both applied to the worms take up more as compared to only adding one element. And we could identify a specific role for the metallothionein 1. And we did not see any neurodegeneration or any oxidative stress. That's why we hypothesize that maybe the metallothionein 1 as a binding protein binds the manganese, that it's not active anymore, and therefore it's maybe a potential protective modulator. I could further show you the importance for looking at the genetics and what a genetics might impact. So genetics impact the whole homeostasis that we should definitely think about. Maybe we need recommendation of trace elements for specific diseases, for specific patients, because it might be they might have an altered trace element homeostasis. So you see, we have a lot of gaps of knowledge, but I hope I could convince you that we have to work on that in order to give better trace element recommendations for age-specific and also disease-specific patients. With this, I hope I could give you some interesting insights. I want really to thank all the collaboration partners, especially Merle and Jesse, who produced a lot of this data I showed you today. And a special thanks goes to my Brazilian partners, friends. I don't know how many years we know each other now. Thank you so much. Priscilla, Diana both contributed to the manganese studies. Thanks a lot for being amazing collaboration partners, and thank everyone else for funding. And I'm happy to take any questions you might have.
Great, Dr. Yulia. That was a brilliant lecture that you bring us today. Our attendees are really excited for this lecture, and they have addressed some really good commentaries. And I think that Dr. Diana and Hugo have some commentaries too to address.
Can I start with some questions?
Yeah, for sure.
Okay. So thank you for the great talk. It was really, it's fascinating. And I didn't know much about trace elements, and I think it's very, very insightful. And it's clear that we need more work and everything. There's a couple of things that I wanted to ask you. One thing that caught my attention in the first set of the data you showed was the concentration of manganese. Because if I recall well, when you measured the amount of oxidative stress in, I think, reactive oxygen species or something like that, it seemed to me that the highest concentration of manganese, which was about 100 millimolar, was actually giving less of an oxidative stress than lower concentration, as if you had an optimal thing. And then when you go on to the next part of the manganese phenotype, it seems like you're using higher concentration, like 250 millimolars. So how do you reconcile this concentration discrepancy? Maybe this could be explained. So that's the first question. I'm going to give you two questions. And the second question I have more related to the later part of your talk about the methylmethionine thing, which I think is of course very interesting. And maybe you mentioned this, maybe I missed it, but what happens if you provide more manganese and zinc combined in a worm that lacks the methylmethionine? Would you then have an increased uptake as well, or would you not see something, or would you have an effect on lifespan maybe?
So I start with the first question. So it's a very good question, because you recognize definitely what is like the problem with the dye assays. So with the dye assay in the alive worms, the DCF assay, we just plate the worms and then we are kind of measuring for a long time. And normally we expose the worms only for 1 hour or 4 hours. And here we expose normally the L4 stage worms for 1 hour, and then you have an LD50, so 50% of the worms are killed by manganese, but it's about 250 millimolar. That's why all studies are done with 250 millimolar and less, 150 millimolar. And only the DCS assay, because they are plated and you can like measure this for a longer time, we measured for a longer time. But the issue is, in case they are staying longer at 100 millimolar, they also start to die, and then you see the decrease in the ROS, because you see this is always a problem in this assay. This is why you normally have to compare only the 1 hour to all the other assays. I just showed it because it's, you know, it's nice to have, you know. And the second questions, we took mutants lacking metallothionein 1, and we saw exactly what you were predicting. So we didn't see any effects on lifespan, because the metallothionein mutant already has a huge impact on lifespan. So it's like almost the normally we have like a 12 days for the LD50 on lifespan. This has only 5 days. And in the mutants, we found differences in the uptake. So here we could not observe anymore that they are taking up more in case both elements are present in comparison directly to like the one element. This is why we also think that metallothioneins has two orthologs in C. elegans, metallothionein 1 and 2. And we think that 1 is the responsible form because we do not see a lot gene expression in 2, and also the worms are not behaving very differently.
Okay. Very nice. Thank you very much.
You're welcome.
Amazing. So good evening, everyone. Good evening, Julia, Elias, Hugo. Julia, thank you so much for the great talk. You know that this is a topic of my particular interest, so it was amazing to listen to you again. Thank you for coming here, for giving this great talk. And
thanks for inviting me.
No, it's amazing, and thank you for all your collaboration with our groups. It's invaluable. It's amazing. So thanks a lot, and thank you.
You're
for the
always welcome.
Thank you. So you know, I have a particular interest about manganese and iron toxicity, and you know that we've been I've been sending samples so you can measure metals for us. And basically nowadays we are very interested about manganese and iron and their neurotoxic effects. So I looked at your data about DNA damage. It's pretty impressive. And I don't know if you look at this DNA damage when you combined manganese and zinc exposure.
No, we haven't yet. So it took us, to be honest, several years to get all of this work done, because looking at DNA damages in the worms, you know, I was starting with this, you know, I'm always in a good mood. I said to the student, yes, we are doing this. This will work great. 3 years later, I'm a bit more clever now. I have a very talented PhD student, and it took her more than 2 years to get any like pure DNA that is not destructed. We have only 6 chromosomes, so we have a lot of things we had to struggle about, like doing DNA damage response answers, stuff like this in C. elegans. So we have not combined yet. This is like the plan in the future, but we have now validated all stuff with positive controls, and we have only applied manganese in the beginning, because we also had to evaluate different positive controls, since all of the positive controls are causing different amounts and kinds of DNA damages, and we wanted to see as much as we can before applying the method for the metals.
So, you know, there's a group here in Porto Alegre, and they validated a method for genotoxicity for comet assay in C. elegans. So I don't know, maybe we can share that, and maybe you guys can see if it's for your studies as well, because I noticed that they found very interesting results looking at the genotoxicity. And then there are some other guys. Yes, and I know there are guys in Germany that are also working with genotoxic effects, and maybe we can share those protocols.
That would be amazing, since comet assay is quite complex in the worms.
Yes, exactly. It's not a single cell. It's like a whole worm comet assay. So let's see how it goes. And my second question is, you look at the metallothionein expression when you combine manganese and zinc exposure, but did you look at the divalent transporters or ferroportin expression? Did you look at those?
We have looked at all of them. So we have looked at SMF3 as all of the manganese importers. We have looked at all the zinc importers. And so we studied about more than 20 transporters. And so, for example, the SMF3, the DMT1 ortholog, it increased with manganese, but it also increased the same amount with manganese and zinc. So all of the importer data were not giving an explanation why, in case manganese and zinc are present, more is found in the worm. So the level of being increased were about the same.
Did you look at the exporter, ferroportin?
Yes, also ferroportin was only a little bit upregulated, but not as much that we could really show that this might be the reason for our findings.
Okay, all right. Thank you. We can talk later.
Can I ask? Sorry, because it's something that so I used to work on aging, and so your data is interesting because you show on one hand that you have a significant increase in oxidative stress and damage to DNA, but you do not have an impact on lifespan, and so that kind of contradicts some of the theories, at least. Could you comment on this?
Yeah, but this is kind of it's a very good question. It's also very hard to compare. So the first studies with the manganese oversupply are done in liquid. So we have done them in absence of E. coli, just for, a lot of manganese for a short time. This is causing oxidative stress. Later on, with the combination studies, we decided, because we wanted to see aging-specific effects, to enrich the E. coli. And you already recognize that here also the manganese amount only were very... Okay, for cell culture people, it's still high, but it's lower as compared to the acute dosing in the beginning. And so we have two different kind of feeding protocols, to be honest. And in the second feeding protocol, with lifelong little amounts of manganese, does not cause oxidative stress that much, only very slightly we could identify here. And I'm not sure if we have any counter-regulating mechanisms, that we do not really see anything on lifespan. Maybe also due to the time, we have an upregulation of the metallothionein, maybe a manganese binding, that we do not have a reactive form for a Fenton-like reaction anymore. This is, I don't know yet. This is a very good question, and we definitely need to answer that in the future.
Okay, great. Thank you very much.
Thank you very much, Dr. Julia. We have some questions and commentaries for you. I would like to address some, not a question now, but a commentary, a particular commentary that was addressed by Beatriz Harumi, that she said that such a beautiful and wonderful presentation. They speak into even I, that I'm not fluent in English, could understand. So, thanks, Dr. Julia, to make it possible. Another question that we have for you is by Maria Cristina Vasconcelos. She said, Dr. Julia, do you think that it's possible to use elegans to analyze heavy metals?
Sure. I think the best person here in the room is Diana for answering that. So she's applying a lot of the real toxic metals. I know we have done some data on mercury, but for us the mercury is more positive control because it's very toxic. But we also have data on arsenolipids or arsenic species. We have done a lot of things together with Diana regarding lead, mercury, and stuff like this. So you just need to check. So you're still in a model. So check the limitations of your model. And once you see that your signaling pathways you want to study are conserved, you can feed the worm with whatever you can think of.
Okay, thanks. Another question by Gabriel Vico, I believe that is Gabriel. He said, high manganese levels led to manganism, who is like Parkinson's disease. But what happens in the opposite, low manganese levels? In the opposite, low manganese levels.
So low manganese levels we only know from animals. And here we have in cattle that they are kind of a bit dizzy and the tongue hangs, they're getting tired, stuff like this. But in humans, this is not really existing because a deficiency has never been observed. Since everywhere you get manganese in your drinking water, in your normal diet, that's why normally a deficiency is non-existing.
Okay, another question by Josiane Mann. She said, great, congratulations for your work. I would like to know if you analyze the toxicity of manganese with any cell death event.
Cell death event like apoptosis?
I believe it so.
So we have checked for the manganese overdosing in the beginning. We have checked if we can see more apoptotic corpses, and we couldn't identify apoptosis in the germline cells. Because this was one of our hypotheses, in case manganese is causing DNA damages, the cell might decide for apoptosis, but we could not identify any apoptotic corpses. We cannot explain that yet, so in case you have any great hypothesis, I'm happy to take and to try in lab.
That was one of my questions that I was thinking about your presentation, that you've said about you've said about the genomic stability that the L1 to L4 phases has, and so I'm starting to think about piRNA's influences in that may be a possible pathways with transposable elements that are usually involved in this genomic stability, but I would give it these insights. So we have another question made by Marcel Valandro. He said, hi, Dr. Julia, it's a curiosity about this MTL1 enzyme response. Is just only to divalent metals, or in different metals these homeostasis too?
A very good question. So right now there is not a lot knowledge about metallothioneins in worms. It's not known yet how they are also be regulated. One of my collaboration partners at King's College, Stephen Stürzenbaum, maybe some of you know, he works a lot with the metallothionein worms, and I got the worms from him. And he works with zinc and cadmium and compares here effects of zinc and cadmium regarding the metallothioneins in the worms. This is the only other metals I know which are definitely binding to metallothionein, but I'm not sure if there are any further studies. This I definitely then need to check.
Okay, so thank you, Dr. Julia Bornhorst. That was the last question. We are hugely thanked for your presentation, and we hope you to visit us someday here in Recife.
Hope so too. Thank you so much, Elias. You did a wonderful job.
Thank you.
Promising this to me.
I promise this already. In case we would not be in a pandemic situation, I would have come last year.
Oh, thank you so much, Julia. I received right now some pictures from high school students watching your presentation from here from Engenho do Meio. We have a project over there. It's a community in Brazil, so in Recife. And we are so happy. Thank you guys for the picture. I just received it here. Thank you so much.
Once you have like questions later on, do not hesitate to contact me. Just write me an email. And Google Translate can also translate Portuguese.
Okay. Thank you so much, Julia.
You're welcome.
I hope you see you soon. Okay, we see you soon. Thank you. Bye-bye.
Bye.
Thank you,
So
bye.
now we have another lecture with Professor Brian, who is already here. Hello, Professor Brian.
Can you hear me?
Yeah.
Excellent. Thanks for having me. I really appreciate the opportunity to talk about what we've been doing here the past couple of years.
Thank you so much for being here. And we have here Professor Diana, Professor Hugo, and my student Elias, who will introduce you this night.
Oh, great.
Thank you so much.
Good evening, Dr. Brian Kramer. It's such a pleasure for me and for Laes to have you here. It's actually an honor to have you here. And so I'm going to present you to our attendees. This is Dr. Brian Kramer. He's going to talk to us about using C. elegans to investigate molecular mechanisms in Alzheimer's disease and related disorders. During his Bachelor of Science, the Dr. Brian Kramer studied biochemistry in Western Washington University. But in his PhD, he also studied biochemistry, but now in the University of Wisconsin-Madison. Currently, he is a researcher and professor in the Division of Gerontology of Geriatric Medicine of University of Washington in the US. His research explores how protein aggregation contributes to neurodegenerative diseases, including Alzheimer's disease, amyotrophic lateral sclerosis, and frontotemporal degeneration. Welcome, Dr. Brian Kramer, and we hope it's going everything well.
Thanks so much. I appreciate the kind introduction. So first off, I would like to thank Priscilla and the rest of the organizers of this meeting for giving me the opportunity to talk about our work using C. elegans and other organisms to study the molecular mechanisms of Alzheimer's disease. So can you see my slides?
No, we are not. Yeah, now you've seen. Thank you.
Okay.
Yeah.
Perfect. So I'm going to try to convince you today that our C. elegans models have told us some things about Alzheimer's disease that are very much disease relevant. So this is going to be a translational talk, starting with C. elegans and moving into mammalian systems. Let's see here if we can get my slides to work. Okay, so this is my team, and as I'm sure you all know at this point, science is largely a team sport. And so these are the people that have contributed to the work. And our collaborators and funding agencies. None of this work would be possible without all these people and agencies helping us out. So in my lab, we're focused on neurodegeneration. Specifically, we care about neurodegenerative diseases such as Alzheimer's disease and some related disorders like progressive supranuclear palsy and frontotemporal lobar degeneration. We also focus on a motor neuron disease called amyotrophic lateral sclerosis. I'm going to talk to you about these neurodegenerative diseases, mostly in the context of trying to understand at the neuronal level what's going on. So, here's the outline of my talk. It's going to be given in 7 parts, starting with C. elegans and then progressing into mammalian systems to try to understand what's going on. So we're going to focus on tauopathy disorders for a big portion of this talk. And tauopathies are brain disorders with abnormal tau deposition. So Alzheimer's disease is probably the most prominent tauopathy. It is characterized by the presence of amyloid plaques and neurofibrillary tangles. Amyloid plaques are made of the A-beta peptide and occur outside of neurons, and the tangles occur within the neuron and are composed of a protein called tau. And so this is the reason why we're so focused on tau, is that essentially the neurofibrillary tangles in Alzheimer's disease and related dementia disorders are a polymer of abnormal aggregated tau. This is a beta-pleated sheet structure that is diagnostic of the neuropathology of Alzheimer's disease. Many other dementia disorders also exhibit tau pathology, and in Alzheimer's disease, it's thought that the amyloid plaques may initiate the disease cascade, but it's actually the neurofibrillary tangles that kill the neurons. So in addition to its pathological role, tau has a normal function in neurons where it stabilizes microtubules in the long axons of the nervous system. And it's only when tau gets into trouble and begins to aggregate that we see problems where the microtubules begin to fall apart for lack of functional tau. So for quite a number of years, it was thought that amyloid plaques started the disease process, and tau is just a marker of neurodegeneration and not really participatory in the whole process of neurodegeneration. But some studies at the turn of the last century, that is the very early 2000s, late 1990s, showed us that actually molecular abnormalities in the human tau gene could cause disease. And that is that familial forms of frontotemporal lobar degeneration are caused by mutations in the gene encoding tau. And these mutations alter the biochemical microtubule stabilizing properties of tau, and they alter the tau aggregation kinetics. And this alone is sufficient to cause dementia in the absence of any amyloid pathology. But even in Alzheimer's disease, where amyloid is present, it's been well studied for quite some time now that the degree of burden of tau pathology, that is, as the Braak stage increases, that's more widespread tau pathology, we get more severe impairment in cognitive function. And this is not the case for amyloid. So it very much seems that the burden of tau pathology is dictating the tempo of neurodegeneration in Alzheimer's disease. So that's just a little background on the disease. What we're really trying to understand is how we might go about finding protective variants that will help us understand disease mechanisms and potentially develop therapeutics. And you may think that's a little bit of an unusual thing to think about at this early stage in understanding the disease process, but I think we've learned some lessons from studying heart disease. And in heart disease, focus on protective variants has allowed us to very rapidly find new therapeutic targets. And in the case of heart disease, it's been shown that drugs with genetic support are more than twice as likely to be successful in clinical trials. And so what was seen in heart disease is that there's a gene called PCSK9, and it was shown that loss of function alleles in humans were highly protective against heart disease, while gain of function alleles in humans caused a severe earlier onset form of heart disease. And so this suggested that controlling PCSK9 levels might be a way to protect against heart disease development. And so we went from a genetic discovery in humans to several approved therapeutic drugs targeting PCSK9 in less than a decade, which is really very lightning fast for drug development. And unfortunately, in the genetic analysis of Alzheimer's disease thus far, we have not really found very many strong protective variants that are actionable. And so we've decided to lean upon the power of C. elegans genetics, which I'm sure many of you are familiar with, to try to see if we can find protective variants using C. elegans. So in my lab, we take a translational approach to study tau pathology. We have human tau transgenic C. elegans, which I'll tell you more about in a little bit, is our primary discovery platform. We do a lot of genetic and small molecule drug discovery type screening in C. elegans, and then try to validate interesting findings from the C. elegans model in simple cultured human cell models that overexpress human tau and have lots of aggregates. And then hypotheses or therapeutic ideas that have been validated in C. elegans in this human cell models, we will try to translate into transgenic mouse models that overexpress human tau and get a severe neurofibrillary tangle type of pathology. So you may wonder why one would start with worms to study human disease. I think many of you already know the answer, but the simple answer to this question is for the experimental advantages, which are many. I think probably most importantly, just for studying aging-related human diseases, is that C. elegans age relatively rapidly compared to humans. They have a 3-day egg-to-adult life cycle. They live about 20 days in normal culture conditions for wild-type animals. So that's very useful in studying the effects of aging. Whereas in mice, you're looking at years. In higher organisms than that, you're looking at potentially decades to look for effects of aging. So this allows somebody as impatient as I am to rapidly address genetic questions related to neurodegeneration. Also, the worm has a number of other distinct advantages. There's a neuronal wiring diagram where the connections between all the worm neurons at the synaptic level are known and have been mapped at the EM level. There's an invariant cell lineage that's been well studied. They have relatively simple behavior. They're optically transparent, so we can look inside of them as they're alive and see what is going on in the various molecular processes that we're interested in. And we have a bunch of powerful tools for transgenesis and genetic manipulation of the genome, including classical forward genetic approaches as well as reverse genetic gene targeting type of approaches using CRISPR and such methods. So quite some time ago now, we developed a C. elegans model for human tauopathy, where we've taken human tau encoding cDNAs, hooked them up to a pan-neuronal promoter, in this case the H3 promoter, and driven expression of this tau cDNA in all worm neurons. And we've done this also in the presence of a marker transgene that lights up the pharynx in green, so we can very easily see our transgenic animals from non-transgenics. And we've taken these transgenes and integrated them and backcrossed to clean them up, and so we've got a multi-copy overexpression scenario where we drive high levels of tau expression in transgenic worms. And for those of you that have looked at worms before, this is the phenotype. We've got two movies here. On the left is an N2 wild-type animals crawling around the plate in a smooth sinusoidal waveform. On the right, we have tau transgenic animals. And while they're not fully paralyzed, clearly they have uncoordinated locomotion. They kind of drag their tail around. And as we'll get into this, this is caused by a neurodegenerative phenotype that they have. So let's see, there's the next slide. So this tau transgene causes a number of defects, most obvious being a progressive uncoordinated locomotion phenotype. They also have a shortened lifespan, shown down here, some lifespan down. Here's some pictures of different worms. These are non-transgenic worms. These carry wild-type tau here in the middle, and then we have two different mutants carrying a V337M mutated version of tau, or a P301L mutated version of tau. Both these mutations cause early onset frontotemporal dementia in human populations. So the tau transgene causes uncoordinated phenotype, where the tau transgene with mutants causes that to be worse. Substantial biochemical studies that we've done over the years have basically shown that both wild-type and mutant tau are toxic. The wild-type tau is less toxic, and this is probably because it has a higher critical concentration for aggregation than the mutant tau, but you can get basically the same effect with 3- or 4-fold more wild-type tau than mutant tau. You get a progressive behavioral phenotype that is accompanied by accumulation of insoluble tau that is hyperphosphorylated. This leads to degeneration of neurons, in particular axonal degeneration, frank loss of neurons, lots of amorphous tau aggregates. And when we look in these tau worms for any given concentration of tau, the presence of these human disease-causing mutations make this more severe. What we don't see in this worm models, we don't see mature neurofibrillary tangles that one would see in Alzheimer's disease. And we think this is because it takes a fair amount of time, probably months to years, for neurofibrillary tangles to form, even under the most adverse conditions in a mouse. It takes several months. So we just don't think the worms live long enough to see neurofibrillary tangle formation. So like I said, we published this quite some time ago. You can, of course, read the articles if you want, describing the model. But what we have done with this model that I think is very informative in terms of finding protective variants is taken a classical genetic approach where we take C. elegans that have this tau uncoordinated phenotype that's very severe, expose them to mutagen, and then look for homozygous suppressor mutations, or recessive homozygous loss of function suppressor mutations that have restored locomotion in the presence of the tau transgene. And we've been doing a fair amount of this over the past several years. And one of the genes that initially came out, and the gene that we're going to spend a fair amount of time talking about today, is a gene called SUT2. This stands for suppressor of tauopathy 2. And this gene, here's again the tau transgene. So we've got the non-transgenic, they move normally. The tau transgenic obviously compromised in their locomotion. And then we've got SUT2 mutant with that tau transgene. You can see they move pretty well. They're not quite back to normal, but they're pretty close to normal. So they're highly resistant to the toxic effects of tau. And we've used this approach and some other approaches to identify a bunch of protective variants that modify tauopathy. And some of these are published, some of them not. We've got genes involved in the dopamine synthesis pathway, particularly dopa decarboxylase, that we've shown through reverse genetic approaches modify tauopathy. Small molecule screen has pointed the finger at D2, D4 receptors. We found the master regulator of the unfolded protein response, XPP1, can protect against tauopathy. SUT2, which I just showed you, we're going to spend some more time with here in a minute. We'll come back to. We recently found a new SUT called SUT6, which I'll hopefully be getting that story out. Another family of genes that are all related that came out of another SUT forward screen. Found some tau kinases, tau phosphatase, and then some other proteins involved in RNA metabolism that we'll talk about some more. So there's a bunch of protective variants that we can identify using C. elegans, but I think, at least for my lab, and I think for maybe the field at large, it's been important to establish for the field that these C. elegans neurodegenerative disease models are really telling us new things about what's going on in the mammalian brain when neurodegeneration is happening. And so we really want to focus the next part of this talk on what is the mammalian homologue of SUT2 doing? Is it involved in tauopathy, and what can it tell us? So, we showed that knocking out SUT2 protects against tau pathology in C. elegans, and that we wanted to really address what is going on in mammals as well. And so let me tell you just a little bit about SUT2 and what we know about its protein architecture. So in the genomes of most metazoans, probably all metazoans, certainly the ones that have been sequenced, there's a single SUT2-like homologue, which is, I think, defined by its C-terminal zinc finger domains, which have this very specific CX5, CX5, CX3, and CX5, CX4, CX3 zinc finger arrangement. And that's conserved in both C. elegans and mice and humans. Also in the protein, we find an N-terminal PWI-like domain, which is called the SUT2 N-terminal homology region, and both a nuclear localization and nuclear export signal, which suggests they can shuttle back and forth between the nucleus and cytoplasm. So one of the first things that we did, when we initially cloned SUT2 and started to study both it and the human protein MSUT2, was really, there was no publications, there was nothing known. And so we raised antibodies against both proteins, both the human and the worm, and started to ask questions, all right, where does this stuff get expressed in human cells and in worm neurons or other worm cells? And what we saw, shown here, is a worm nerve cord, and you can see individual neurons. The nuclei are lighting up with these what appear to be puncta or speckles that contain SUT2. And when we stained human brain, this is a human hippocampal section, again, in the nuclei of neurons, we see speckle-like appearance. And in fact, when we stain human HEK293 cells, these hex cells have MSOT2 present in the nucleus as well. And if you go and look for what are nuclear speckles, turns out there is a structure called the nuclear speckle, and it is a domain of the nucleus that is enriched in splicing factors and RNAs. And it is marked by the canonical monoclonal antibody called SC35, which actually recognizes a protein called SRM2 that we'll talk about a little bit more in this talk. But we've showed that MSOT2 and worm SOT2 seem to be in nuclear speckles within the nucleus. And so that's a little peculiar to us. I mean, tau is mostly in the cytoplasm. There's some talk in the literature about there being some nuclear tau, but there's certainly not very much of it compared to how much is in the cytoplasm of neurons. But I think for the field and for our satisfaction, the thing that we most wanted to understand was, does MSOT2 in mammalian systems, when it's knocked out, protect against tau pathology, just like it does in the worm? And so we set about making a mouse where the MSOT2 locus was knocked out. And to do that, we used the knockout mouse consortium comp allele for MSOT2 that basically makes an allele very similar to some of the SOT2 alleles where we have truncated the protein at these zinc fingers. And so we made this mouse, and the homozygous, and bred the allele to homozygosity. And the homozygous knockout makes no MSOT2 zinc fingers, and the heterozygote has a half dose. But to our great pleasure, we learned that the knockout mice were viable. They seemed to be healthy. They were at least somewhat fertile, and they seemed to be cognitively normal and neurologically normal. We did notice that female mice gave relatively poor maternal care for their young, which is of concern, but it was not preventing us from doing the experiment. So we took these MSOT2 knockout mice that are very alive and crossed them to another mouse line that expresses severe neurodegenerative driving transgene. These are called the PS19 mice, and they overexpress human tau carrying a P301S mutation. And this P301S mutation has been shown in families that have frontotemporal dementia to cause a very severe early onset form of tau-only dementia. And the mouse model seems to be a very faithful representation of that human dementia. They have early tau hyperphosphorylation, severe and progressive loss of synapses with a lot of neuroinflammatory change. This causes them severe cognitive memory dysfunction. They get a lot of tangles. They get progressively worse, and they eventually lose neurons in great numbers. And this tauopathy kills them quite young, at about a year of age on average. So this is a relatively aggressive tauopathy model, similar to the worm model in that it is quite aggressive. And we crossed our MSOT2 knockout to these tau mice and asked the question, what happens to tau pathology? And so shown here are some brain sections from the hippocampus in a PS19 mouse and in a PS19 mouse that has MSOT2 knocked out. And what you can see is that while there's tau pathology in both brain sections, and this by tau pathology we're talking about phosphotau, which is one of the early markers of tau pathology, there's definitely visibly less of it in the MSOT2 knockout mice. When we look at more advanced pathology, that is the pretangle conformations of tau, it's thought that first you have phosphotau, then pretangles begin to form, and then ultimately tangles. We also see a decrease in the pretangle conformations of tau, again looking at the hippocampus, although we see this in other brain regions as well. And then finally, the mature pathology in Alzheimer's disease and related disorders is the neurofibrillary tangle formation. And the PS19 mice tend to get lots of tangles in their hippocampus. And in the MSOT2 knockout mice, there's almost no tangles at all. So they're very well protected against neurofibrillary tangles. When we look at other readouts of neuropathology, for instance, neuroinflammatory change, we see that they exhibit much less astrocytosis when MSUT2 is knocked out. So the PS19 mice have lots of astrocytosis as detected by GFAP staining, but the MSUT2 knockouts have much less. Likewise, when we look at neuron loss, shown here is the hippocampus stain for crescent violet, which in a normal mouse shows a nice thick ribbon of neurons in the CA1 region. And here, in the PS19 mice, that's very thin, almost gone. Whereas in the MSUT2 knockout mice, we see protection from the tau pathology, not nearly as much neural loss. We then looked at behavioral readouts using the Barnes maze, which is a spatial learning and memory task. And we can see that the time to escape the maze is really something that mice should get better at with time. And these MSUT2 knockout mice do get better with time, but the PS19 mice on their own really don't learn the task like they should. Furthermore, when you test the mice after they've learned the task by hiding the escape hole, the MSUT2 mice do a very good job of remembering where that escape hole should be, whereas the PS19 mice aren't nearly so good at remembering. So just to summarize the mammalian data in relation to the worm data, in a PS19 mice, we have, in tauopathy conditions, we have tau coming off of microtubules, aggregating, forming neurofibrillary tangles, which cause the neurons to die and provoke a lot of neuroinflammatory change in the process, which can then spread to other neurons, leading to additional killing of neurons. When we knock out MSUT2, the tau no longer is aggregating, we don't get neurofibrillary tangles, and the mice are protected. This is very similar in mice to what we have seen in worms. Now, I'm not going to spend the time going over the data because we're already starting to get a little short on time, and I have a lot more stuff about mechanisms I'd like to tell you. But when we overexpress SUT2 in the worm, we see that in the presence of the tau transgene, everything gets worse. In the absence of the tau transgene, SUT2 overexpression has no effect. And in the mice, we see the same exact thing. Loss of MSUT2 is protective, while overexpression of MSUT2 drives additional tau pathology. Okay, so now we're going to turn our attention to what might the molecular mechanism of this neuroprotection be. And so initially, when I was naively approaching this problem, I was like, MSUT2 is a zinc finger protein. Many zinc finger proteins are transcription factors. Therefore, I think that knocking out MSUT2 is going to lead to a lot of transcriptional changes. And so we'll just do some brain-focused RNA-seq, and that'll tell us, what are the important genes that matter. And much to my surprise, when we did this RNA-seq experiment, we saw almost no genes that had even a twofold change. The only one that was exceeding twofold decrease was actually the messenger RNA for MSUT2. So that was pretty deflating initially for us. But we went back and remember the data I already showed you before, that MSUT2 and SUT2 are expressed in nuclear speckles. And I thought, well, maybe that will tell us something about its function. So we asked a simple question about what other proteins does MSUT2 interact with? And we did a yeast-2 hybrid screen and then some biochemical validation. And the thing that came out of that screen over and over and over again was a protein called PABPN1. And so we showed that, biochemically they interact, in yeast-2 hybrid system they interact. When you IP from mammalian brain and cell culture lysates, MSUT2 brings down PABPN1. So what is this PABPN1? PABPN1 stands for poly-A binding protein nuclear 1. And so this is an RNA binding protein that binds to the poly-A tail of messenger RNAs. It acts as like the ruler that measures the length of the poly-A tail, and a processivity factor that recruits the poly-A nuclease to the cleaved messenger RNA to make it have a mature poly-A tail. Interestingly, PABPN1 also in its own right is a disease protein in that expansions in its polyalanine tract at the N-terminus can cause a disease called ocular pharyngeal muscular dystrophy, which has protein aggregation of PABPN1. Interestingly, these ocular pharyngeal muscular dystrophy patients also tend to develop dementia. All right, so we then asked the question, well, does MSUT2 seem to be where poly-A RNA is? And the answer to that question is yes. So here's a stain for poly-A RNA, and here's a stain for the co-localization of poly-A and MSUT2, and we can see they're in the same place. So then we wanted to ask the question, is MSUT2 really in speckles, in these nuclear speckles? Is it really together with PABPN1? And so we did some high-resolution SIM microscopy, and we showed that MSUT2 co-localizes with PABPN1 in the nuclei of hex cells. So shown here is MSUT2 staining, very similar looking PABPN1 staining in yellow. That's the co-localization, where MSUT2 and PABPN1 are both in green. They show a constant of co-localization of 0.88, so they're highly overlapping their expression pattern within the nucleus. A second assay using proximity ligation also showed that MSUT2 and PABPN1 are right on top of each other in the nucleus in hex cells. So we then, people have known for a long time that PABPN1 binds to poly-A RNA with a relatively high affinity. So we used a technique called surface plasmon resonance to measure the affinity of PABPN1 and MSUT2 for poly-A RNA, and we found the dissociation constant for MSUT2 to be about 60 nanomolar, whereas for PABPN1 it's more like 240 nanomolar, so it's about fourfold more avid than PABPN1 for the poly-A RNA. So it's definitely a poly-A binding protein. We then asked the question, well, what happens in human cells when we knock out MSUT2 or PABPN1 to the tau pathology? So we have this human cell line that I talked about earlier. This is the HEC293 cells that overexpress human tau, and you can see in green they're stained with AT180. When we knock down MSUT2, that phospho-tau goes away mostly. When we knock down PABPN1, we see a dramatic increase in the levels of this phospho-tau. We see similar effects for pretangle conformations of tau that are stained by either MC1 or TOC1. So this seems to promote tau aggregation. Now at the same time we were doing a bunch of this work trying to figure out what MSUT2 and PABPN1 have to do with tau, there was another group, Anita Corbett's lab at Emory University in Atlanta, Georgia, that was studying MSUT2, also known as ZC3H14. And they were showing that MSUT2 and PABPN1 have opposing effects on poly-A tail length. That is, when you knock down MSUT2, poly-A tails get longer, and when you knock down PABPN1, poly-A tails get shorter. So we've shown that MSUT2 and PABPN1 have kind of an opposing relationship, both for polyadenylation and for tau aggregation, at least in cell models. Okay, so what does MSUT2 have to do with human disease? So we started having, armed with these MSUT2 specific antibodies that see the human protein, we started to ask questions about what happens to MSUT2 in Alzheimer's disease. And the first question that we asked is, where is MSUT2? And it turns out that it's in a lot of nuclei within the neurons of Alzheimer's patients, but it is in also the nuclei shown in green here of neurons that contain neurofibrillary tangles. And this flame-shaped neuron is kind of a characteristic morphology for neurons with tau neurofibrillary tangles. So we worked up a number of cases, I think about 30 or so, and found that MSUT2 and PABPN1 show a surprising staining pattern. That is, if you look in normal control cases, you have clearly nuclear, predominantly speckle localized MSUT2 and PABPN1. And we see that also in a subset, approximately half of Alzheimer's cases show that same speckle-like distribution. But in a subset of Alzheimer's cases, there is no MSUT2, and there's very little PABPN1, so they've become depleted. And so we think that this complex of MSUT2 and PABPN1, as we know they can bind to each other, has become depleted in Alzheimer's disease. And so we asked our clinical collaborators, what is it about these cases that are different? These cases that have normal MSUT2, why are they have Alzheimer's disease, and why are they different from these cases with Alzheimer's disease that have a pretty severe depletion of MSUT2 and PABPN1? And my collaborator, Dr. Bird, came back to me and said, Ryan, what's going on here is that these cases that have depletion of MSUT2 have a more severe form of the disease. They have an earlier age of onset, and that age of onset is about, somewhere in the range of 8 to 10 years, depending on, if you're scoring by MSUT2 levels or PABPN1 levels. I think we have a little more power with PABPN1 because it's a more abundant protein. But just to put this in context, an 8-year earlier age of onset is a more dramatic effect than having ApoE4 allele, which is the largest effect size risk factor for Alzheimer's disease. So it's a very, from a genetic standpoint, that ApoE4 allele drives significant Alzheimer's risk. And so this is a similar age of onset effect to ApoE4. So that's a pretty big deal. So the other interesting thing that we saw was that when we looked at these cases that have MSUT2 and PABPN1 depleted, they have a more severe neurofibrillary degeneration. They have more tangles, they have fewer neurons. So that was a pretty surprising result as well. Well, maybe not surprising, given that they have a more earlier onset, they may have a more severe disease. But it was striking nonetheless. The surprising result was actually when we looked at the neuroinflammatory change, what we saw is the cases that had their MSUT2 and PABPN1 complex present had a more severe neuroinflammatory response, suggesting that maybe that is protective, while the cases that had their PAB and MSUT2 complex depleted had a less severe neuroinflammatory change. All right, so to summarize the mammalian data so far, the polyadenylation machinery can potentially act as a control point in tau-mediated neurodegeneration. And so normally there's this PAB MSUT2 complex that is present in normal situations. It's also present in a subset of Alzheimer's disease and in PS19 mice. This is permissive of tau pathology and leads to selective vulnerability of neurons. In severe Alzheimer's disease, this tau MSUT2 or PABPN1 MSUT2 complex becomes depleted. There's a very severe disruption of microtubules, severe tau pathology, and a widespread loss of neurons. So this is obviously a very, very bad state of affairs. You do not want to lose both your MSUT2 and PAB. However, if we can selectively inhibit MSUT2, and in mice we've done it genetically, that's protective against tau pathology. Microtubules are stable, there's very mild tau pathology, mild neuroinflammatory change, and neurons survive. And so we want to achieve this state here where we've depleted MSUT2 activity but preserved PABPN1. And so this leads us to the idea that perhaps MSUT2 could serve as a therapeutic target. And so, we've used the worm to identify a new target for therapeutics, perhaps. And we've got some data now that suggests that RNA processing may be involved, and we'll revisit that concept here in just a little bit. But I want to briefly talk about MSUT2 as a therapeutic target. We're getting quite short of time here, but I'll whistle through the last couple sections pretty quick. The big advantage of something like this as a therapeutic target is we've already shown the knockout mice and the knockout worms are totally viable, so it's not essential. We know it's expressed in neurons. It has an activity we can target, that is, it's RNA binding to poly A. It's a novel target. The field is clearly in need of new targets for Alzheimer's disease. And we've validated it genetically by knockout in worms, knockdown in cells in mice, all show that this can protect against tau toxicity. From kind of a big pharma standpoint, there are some disadvantages. It doesn't have an enzymatic activity, which is always, the drug company's favorite target are things with enzymes or, 7-pass transmembrane receptors they like as well. But so it's not enzymatic. It's novel, so we don't know a lot about the protein, so that's a little bit of concern. There's no known existing assays. We know that poly-A metabolism is important, so it does have an important function. Our mouse data would suggest that it has impact on fertility, but that's probably not too big a concern for a late life dementia disorder. The biggest disadvantage is there are no known inhibitors. So we're kind of starting from scratch from a therapeutic standpoint. And so we decided to take a drug repurposing approach initially, which shaves down the amount of time it takes to do drug discovery by quite a bit, in that you confine your initial search to drugs that have already got a previous use, safe use in humans in their history. And so that can shorten the drug development life cycle from, decades to years. Still a long path, but hopefully faster. And so we went back to the biochemistry of MSUT2 and set up an assay where we're going to study this RNA-protein interaction using either human MSUT2 or the worm SUT2 protein that we've expressed in E. coli and purified, and ask whether or not it can bind to poly-A RNA. And so we've taken to using an assay called the alpha assay, which is essentially a bead-based ELISA type of assay. The one bead coated, the donor bead coated with avidin, biotinylated RNA that binds to it. If the MSUT2 protein binds to that, it recruits the glutathione bead. If these two beads are close together, you get signal out. And so we can use this by adding different drugs to see if they inhibit the interaction. And to make a long story short, we got an orthogonal assay that involves a different kind of RNA binding assay. This is called a fluorescence polarization assay. It's also a good assay. We did a screen of the Prestwick chemical library, which is about 1,200 compounds, and we found, 8 or 10 that seem to have, reasonable affinity for, or reasonable inhibition of the MSUT2 RNA binding activity. And so this, I think, has de-risked this project. We're now screening novel chemical space in the hopes of finding really potent small molecules that can inhibit MSUT2 well enough that we might be able to try them in mice. All right, so I'm going to skip the last section other than to say we really think that MSUT2 and some of the other modifiers that we've pulled out recently are important because they're all in nuclear speckles. And I think nuclear speckles are a new area that the tau field has recently become interested in. Both my group and Roy Parker's group have recently put out papers about RNA processing in tauopathy and the importance of nuclear speckles. But I'm kind of already running over, so we'll skip about SRM2 and nuclear speckles. We have a recent paper in active neuropath communications I would recommend that you take a look at. Here's the model figure from that paper, where basically we've shown that phospho-tau accumulation leads to disrupted nuclear speckles, and at least the scaffold protein for nuclear speckles becomes recruited into the cytoplasm and co-deposits with tau. So in my lab right now for MSUT2, we're really focused on trying to develop MSUT2 inhibitors into lead compounds for trials in preclinical models. We're trying to understand the genetic contributions of nuclear speckle proteins to tauopathy. We're trying to characterize SRM2, and we're trying, above all else, to try to saturate this screen that we identified, SUT1 and SUT2 and these other SUTs that I briefly mentioned, so that we can find all the protective variants that are available to us in C. elegans and try to get a better handle on the neurodegenerative changes. I think, the clue we've got so far from these is that it seems like many of them are involved in RNA metabolism and nuclear speckle biology. And so we're trying to understand that better now that we're convinced that that is also a thing that goes on in human disease. And with that, I will conclude my talk, and I'm happy to take questions.
Phenomenal lecture, Dr. Brian Kramer. That was a brilliant, brilliant experience to have you here with us. I personally love this subject. I'm not working with that, but it is something that I would like to work with. I have studied about the NIT1, MALAT1, some long non-coding RNAs are involved with nuclear speckles.
Right.
And yeah, and I would like to ask you about that. There is the paraspeckles region that may sense some mitochondrial damage that the tau aggregation may input. And so, do you plan to also visualize the involvement of MALAT1 or NIT1? That is, there are some long non-coding RNAs in this pathway, in this involvement?
So that's a great question, and yes, we have actually thought about that and done a couple of experiments. Really, not so much on MALAT1, but on NIT1. We have looked, and it seems that NIT1 is mostly adjacent to and not within the nuclear speckles in human cells. And functionally, if we knock down NIT1 with siRNA, it does not seem to impact tau pathology. So we don't think that NIT1 is probably involved, at least in human cells. What I would really like to know is what is the C. elegans homologue of NIT1, and knock that out and see, genetically, because I think that's a much more rigorous way to test the hypothesis that NIT1 might be involved. But so far as I know, the field has not identified a specific long non-coding RNA that has that NIT1 scaffolding function for paraspeckles. Is that right? Am I missing anything on that? That's right? Okay.
Yeah, that's right.
Okay. So, I keep waiting for somebody to tell us which, what the NIT1 homologue is so we can actually, really address that question.
Yeah, and I've been looking on the neuroinflammation result that you bring to us, and MALAT1 has some impact in human cells on not neuroinflammation, but in inflammation directed by RFs or interferon-related genes caused by the cleavage of TDP43 that MALAT1 promotes. So maybe that would be related with your inflammation results.
Yeah, I suspect that, in a subset of Alzheimer's cases, that's true. And the reason that would be is that, about 30% or so of Alzheimer's cases have pretty abundant TDP pathology. People don't talk about that a lot, but it is definitely there. And it's really in those Alzheimer's cases that have a more severe hippocampal pathology called hippocampal sclerosis, where essentially a big portion or the entirety of the hippocampus has been ablated, and all that's left is some TDP pathology, and there's almost no neurons whatsoever. So that's a very striking change. There's very severe neuroinflammatory change there as well. So, I think it's definitely worth looking at. I've got some neuropathology collaborators that are really trying to understand the interaction between tau and TDP and that neuroinflammatory change in hippocampal sclerosis and how all those things fit together. And I'll suggest that to them if they haven't already thought of it. I don't know, but it's a good idea because I don't think that RNA metabolism gets very fair treatment in our consideration of its contributions to neurodegeneration. And it's only recently that I think people have started to pay more attention. But I think that's right on.
Yeah. Thank you. Dr. Ugo, please, yeah.
Yeah, no, so Brian, thank you very much. I think it's an impressive body of work. It's actually it looks like a book of how to use C. elegans to make great discovery and go all the way. It's actually great. So tons of questions. Probably I won't have time to ask them all, but a few things. If I understand correctly, there's a balance between SUT2 and PABN1, right? So if you knock out the two of them, what happens in C. elegans? Do you have a homolog of PBA in C.
So
elegans?
there is, and we really can't do that experiment in any system but cultured cells. So the worm homolog of PABPN1 is called PABP2, and it is essential. And so loss of function of that is dead.
And a hypomorphic allele?
No, not that. Not that's convincing. It's possible, there's million mutation project alleles that may or may not be loss of function, but we haven't really pushed that angle very hard. But I think that's an interesting thing to approach. If we could get some sort of conditional allele, that would be really useful as well. But
yeah, sure. And so I'm sorry, I'm asking. I'm going to give myself the right to ask 2 or 3 questions. Is that okay, guys? I mean,
you can ask as many as you like, as far as I'm concerned.
So the other thing is that your study reminded me a little bit of the A beta work that was combined with the insulin growth factor, the IGF-1 work, where it was very similar. So studied in worms with the paralysis. And then they identify IGF-1 as DAF2, in fact, I think, as an inhibitor, and then translated it into mice. So did you pick up in your screen, did you pick up any of the traditional aging stuff that could have...
Not so far. No age 1, no DAF2 alleles. We're nowhere near saturation yet, so it's possible they're still there. But, DAF2 in particular is a pretty big target, so I kind of feel like we should have seen those alleles. Probably should just go test it directly with one of the popular DAF2 alleles, but we haven't really done that. I always figured that forward genetics would tell us all the most important things, but that's probably something we should do.
And so you did a chemical screen, right? Why this rather than, I don't know, an RNAi screen or CRISPR?
Right. We did an RNAi screen is the short answer to that question. The big problem with the RNAi screens, of course, is RNAi doesn't work very well in neurons. And this is a neuron model.
That's right.
And so we didn't really get any strong suppressors by the RNAi screen at all. And in fact, if you take SOT2, where we know that loss of function is strongly protective, you can't see an effect. So I don't have any confidence. And every time we get a new suppressor, we do the RNAi just to see, okay, could we have seen this one? And the answer has always been no. So I think
that's important.
Yeah, and some of the mutants we're getting out are not loss of function. Some of them are dominant gain of function. We've got a few alleles like that, and some of them are peculiar non-loss of function recessive alleles we don't really understand the nature of yet. So yeah, RNAi hasn't been helpful. Now we're using CRISPR a lot to, when we get a single allele, to make the same allele over again to validate, to make nulls. It's very nice for that sort of thing. We haven't really figured out how to do a good CRISPR screen in C. elegans. In mammals, that's certainly something we could do, but we've been more focused on the worm side for the discovery work. The one thing I will say about PABPN1, though, is we did go back to humans. We managed to get a couple autopsy cases that have ocular pharyngeal muscular dystrophy, which is known to have dementia. They have tau pathology. So we do think, and it's thought that aggregation actually causes a PABPN1 loss of function. It's pretty thin evidence at this point because the number of cases is it's an N of 2 right now. But I think that it's likely true that PABPN1 is a tauopathy gene.
And so I'll ask after that, I promise. But so one thing that is unclear to me is how do you go from the size of the tail, the polyA tail, to the tau phenotype, to the tau aggregating. So maybe this was the part 7 that you were. I mean, you flew through this, so I'm not sure. So is there a mechanistic link between the size of the polyA tail and the tau phenotypes and the aggregations and stuff and the tangles?
So the short answer to that question is no, and I really wish I knew the answer, because that's, one of the things I spend a lot of time trying to figure out is how does this work? And I have some ideas, but precious little data supporting them so far. And, my idea is that changes in the polyetylation affect the way RNA is packaged, the way messages are packaged and sent out of the nucleus to be translated. And we know from some very old biochemical data that a variety of polyanions can seed tau aggregation. And the polyanion that most potently seeds tau aggregation is RNA. And so I think that what is happening under normal circumstances in tauopathy is that RNA can be, tau can have some access, especially under stress conditions, to naked RNA. And when that happens, it seeds tau aggregation. And it starts the whole cascade. Once you have some tau aggregates around, they're very difficult to clear. It normally has a very long half-life on the order of days to weeks. And so it starts an aggregation cascade. And I think what we've done with MSUT2 and some of these other speckle components in general, and I don't really understand how this works, but what I think has happened is we changed the way the RNA is packaged such that tau can't access that RNA nearly as easily. And so when the cell has a little bit of stress with aging or whatever, instead of seeing this RNA sitting around, the tau can't see it, doesn't aggregate, and so you stave off, the cascade that way. But it's very hand-wavy right now. We're, trying to sort out, how this might possibly work. But, that's just, combining our observations from some like 25 and 30-year-old biochemical observations about tau. We've done a little looking for changes in RNA localization in MSUT2 knockdown cells and haven't really seen much. In situs are pretty challenging to do in worms, so we haven't really got that working yet. But that would be another obvious experiment to do. But I think it's
very cool.
You get right to the point there. That is really, I think, the question is, what is the mechanism? How does this work? Translationally, I think have made good progress in that. We can target this in mammals and make things better. And I think we're getting there with the small molecule approach to the point where we might be able to really demonstrate that therapeutic idea in mice someday. Soon, the next couple of years.
Yeah, no, that's amazing. And so I promise that's the last one. Just the very last one. It's a very something that caught my attention. You said that for the small molecule screen, one of the disadvantages is that it was non-enzymatic. And so
yes.
How could you be sure that SUT2 doesn't have any enzymatic activity?
No, that's a good point. So when we purify the protein, we cannot purify the full-length protein. E. coli, at least, does not tolerate it at all. It just chops it up into ribbons. And we get fragments that are like the size of the zinc fingers, and that's it. When we overexpress the zinc fingers, which is the RNA binding domain, that is stable, and we can purify that. And so we don't formally know. It may be that, the C-terminal part of the protein, which doesn't really look like anything else out there, it could have enzymatic function. You're absolutely right. It could do RNA methylation, or it could be, have some sort of novel RNase domain, or, whatever. But, we haven't really been able to tackle that sort of problem. I think probably need a very talented and persistent biochemistry student to come in and try to tackle, purifying native protein for that. And
hopefully it will be Brazilian.
That would be awesome. Yeah, well, that would be awesome. We get this pandemic behind us and we can actually have people visit and stuff. That would be really fabulous.
Well, congratulations. It's great stuff. Really, I really enjoyed the talk. Thank you.
Thanks so much. I really appreciate it.
Yeah, it was amazing, Brian. It was amazing talk, and I enjoyed a lot, and I'm sure that all the audience liked it a lot because we have so many questions. I just want to say that I've been working with some neurodegenerative disease models, but actually working with A-beta models, not with tau models. And we've been working with the interaction of some toxic metals in these diseases. So So it's another world. And something that people always ask me, and I noticed that you showed the data on rodents about the neuroinflammation. But
Right.
in C. elegans, we cannot study neuroinflammation, and
Right.
something that people also always ask me, it's about microglia and the role of microglia. And I think C. elegans has a microglia like system, and maybe you could tell us about it, please.
So I am certainly no glial expert, but I get the same question, and I feel like thus far it has really been one of the limiters for the C. elegans model, is that if you look at the human GWAS data, the genetic data, there's a bunch of innate immunity, microglial genes, things like this that are present, and most of them are not present in C. elegans. So I don't think that C. elegans really does neuroinflammation in exactly the same way. I think astrocytosis one could probably study if you knew enough about the way worm glia work. And I don't think that worms have glia the way mammals have glia. They don't have microglia and astrocytes. I think that the glia are kind of all-purpose, right? So they have astrocytic functions and microglial functions in the same cell. And so I think if we knew what the right intermediate filament protein was to look at, there's probably a GFAP-like intermediate filament that goes sky high when the C. elegans glia are upset. When something's gone wrong in the brain, the astrocytes go berserk and they express all kinds of GFAP. And I bet you there's an intermediate filament protein just like GFAP that does that. We just don't know what it is. And there's a lot of intermediate filaments to look at, so nobody's actually sorted through that. But I suspect that is an achievable situation, that one could actually find the intermediate filament changes. Then you would have at least some sort of glial readout of neuroinflammatory change. There's also some innate immune stuff that I think one could potentially look at. But even in mammals, the markers for neuroinflammatory change are very imprecise and not GFAP is pretty clean. But when you start talking about microglial activation, there's controversy, and it depends on the context you're looking. And if there's invasion of monocytes or other immune cells into the brain, then you don't know all of a sudden, is that microglia, or is that an infiltrating monocyte of some kind that's differentiating to look like a microglia, or what is going on? So it gets very complicated very quick. So I think that there are definitely limitations from studying this problem in C. elegans in terms of neuroinflammatory change. But I don't think that there's no neuroinflammatory response. I think there is one. It's just simplified, and it's distant enough that it's hard to get a one-to-one correspondence between the worm genes and the human genes. But I think there is a correspondence. We just don't know enough about worm glia to understand it yet.
Okay. Thank you. Thank you for your input. And I think Elias, I think we can go to the public questions.
Yeah, we have some questions for you. So Sanjeev asks, great talk, and sorry if I missed it. Have you checked the effect of any other modified pathological tau, like acetylated tau at lysine 274 and lysine 281? If yes, do they have the same pathological effect?
So, all right. We've looked for acetylated tau using Todd Cohen's acetylated tau antibodies, which were polyclonals raised in rabbits. And if you've done much staining with rabbit antibodies, from polyclonal rabbit antibodies on worms, you're probably aware that there's a lot of cross-reactivity issues. And so we were never able to convincingly show any acetylated tau by Western blot, but we were also not able to fully clean up those antibodies, so I think it's complicated. I tried to get my hands on, I'm trying to remember the person's name and I'm forgetting, and that's terrible. We tried to get our hands on some monoclonals, that have been raised, but we have not succeeded in actually getting those to work. Or I'm sorry, getting those in hand to test them. So it's a little uncertain. We did do some mutagenesis to tau just to see if there was acetylation. I think we did in one site, and it had no effect. So we haven't done this exhaustively, but so far it doesn't seem like if there is acetylated tau, it makes that much difference in the C. elegans model. But then again, the C. elegans model doesn't get mature neurofibrillary tangles, right? I don't think they live long enough. So I think that the evidence for acetylation would suggest that it's a very downstream driver of end-stage aggregation. It probably doesn't happen until after phosphorylation. Some aggregation has already begun. But it's a good question, and I'm not sure of the answer. It hasn't been addressed completely. We've thought about it. It's important, but we just haven't really convincingly shown anything that I can, data that I could show you, I guess.
Okay, brilliant. Another question is from Lucas Aleixo. He said, excellent lecture, Dr. Brian. Do you believe that with the advancement of omics and structural biology studies, we will soon be able to discover integrative pathways between different proteinopathies?
I do. I think that, there's really been a revolution with cryo-EM of the fibrillar structures of tau and different tauopathies. And, each tauopathy seems to have its own particular structure, so that's interesting. We don't have the yet for TDP-43. I think that's starting to come for, both amyloid and synuclein. So I think the actual structures are being defined. How long it will be before we can take our worm model and validate that the structures we're forming in those are similar to human disease, I'm not sure. It takes a pretty big chunk of brain right now to do that. So, as far as the omics, yeah, I think that from talking to my neuropathology colleagues in particular, they're getting very good at, all these large data set analyses and nominating things that change, but then they have no easy functional means to test whether they're important or not. Knockout mice, that's a long and difficult road. And, nobody has a good cellular model of neurofibrillary tangle formation or tau aggregation-driven toxicity even. So, I think models like C. elegans and Drosophila really have a functional role to play in validating a bunch of the, of the things that come out of GWAS, the stuff that comes out of omics, all these things I think, are really in need of systems to test, specific hypotheses. And so, obviously that's pretty straightforward to do in C. elegans. Mostly we've been focused on, genetic discovery kind of work rather than hypothesis testing. But the data that's out there, there's just mountains of data that suggest all sorts of experiments one could do. It's a great question.
Okay, thank you so much, Professor Brian and Hugo, as well as Diana and Elias. Brian was a great surprise for us because we started to study about your research and read your papers. And Lucas, my student, who is here also, was very interested in this protein parties. So we contacted you, and you were so kind with us and was open to discuss about our projects. And it was very nice, and kindly sent us your strain. So we thank you so much for this. I hope many Brazilians now know you and can maybe work with you soon. Okay.
That sounds great. I really appreciate the opportunity to talk to you all, and I hope your work is able to pick back up again as we get more and more back to normal, hopefully in the next year or so. And yeah, looking forward to talking more. It's really fun. Thanks for the opportunity. I appreciate it.
Thank you so much.
Thank you, Brian. It was great. Great talk.
Thank you, Brian.
Thank you.
Thank you, Hugo.
Yeah.
Obrigada,
Thanks.
Diana.
Really interesting questions. I really appreciate your thoughtful questions too.
Oh, thank you so much.
It was my honor to mediate this with Elias and Hugo. It was a pleasure to watch Brian and Julia. It was amazing.
Yeah, and for me it was also a honor to be here with you, guys that are brilliant researchers in your fields. I feel hugely thanked by Priscilla to ask me to mediate this session, and I hope that we see each other someday in some place.
Thanks so much. I really appreciate it.
So now we
me
have
too.
the poster presentations. Oh, sorry, Hugo.
No, I just was thinking.
Okay. We have the poster presentations that will be in pitches format. So we have the meet rooms, and so you can go to the rooms to take a look in the pitches. Okay. Right. Thank you so much to everyone, and see you soon in the final presentation, in the ending of the event. Okay. Bye, guys.
Bye-bye.
Bye-bye.